Hi, my name is Mackenzie Williams. Today I'm going to be presenting on the anatomy and enzymology of fat digestion and absorption. So to start off, we have the initial digestion of fats. So initially, hepatocytes in the liver will secrete bile, and we have the visualization of bile up here at the top. Um, Bile is made up of three different things. First, we have bile salts, and the bile salts are what will facilitate the enzymatic digestion of fats. Secondly, we have the bile pigments. The bile pigments are basically a waste product of hemoglobin degradation, and then we have cholesterol, which will be excreted into the feces. Um, you can see here that you see the initial creation of bile in the liver, and the bile will then move into the gallbladder. This is through the hepatic ducts, so the gallbladder will basically store and concentrate all of the bile until it's ready to be sent into the duodenum through the common bile duct. And into the duodenum here, the beginning of the small intestine, is where the bile salts will begin to break down the fats initially. So one of the main complications with fat digestion is that fat is not water soluble. So as a result, we have to find a different way in the body to break it down as opposed to carbohydrates and proteins. So here we see a large fat globule. This is what fat looks like when it's entering the small intestine and leaving the stomach. So it's pretty large. It doesn't have a lot of surface area for enzymes to begin to break down this fat. So to combat this, bile from the gallbladder will enter the small intestine, and the bile salts will actually begin to break down the fat into smaller emulsified fat droplets, which we see up here at the top visualization. Um, After we have the bile salts begin to break down the larger fat globules, we then have lipase, which carries out the rest of digestion. Lipases will begin to break down fats in the stomach. About 1/3 of digestion of fats occurs in the stomach, but the rest is in the small intestine. Um, so when we have lipases interacting with these fat molecules, you can see that the lipases will remove two of the fatty acids from each of the triglyceride molecules. So then what we have left here is two free fatty acids and one monoglyceride. Um, Additionally, the lipases cannot penetrate bile salts. So we have colipases, which are secreted by the pancreas, come in. It helps to displace some of the bile salts so that lipases can move through and enter, beginning to break down those smaller fat molecules. We have phospholipids, which are digested by phospholipases, and cholesterol are basically just absorbed intact. They don't really need anything to break them down. So additionally, once that those bile salts begin to break down the fats and are broken up by the colipases, they're reabsorbed and go back to the liver to be recycled, which is one point to keep in mind, that we're not creating more bile salts all the time. We can recycle them. So once the fat is broken down into smaller portions, like the free fatty acids and the monoglycerides, they are compacted and formed into small dish-shaped micelles. The micelles then enter the aqueous layer of the brush border. So you can see here a visualization of the micelles. You have bile salts, phospholipids, fatty acids, cholesterol, and mono and diglycerides. This kind of moves the fat portions all together towards the brush border to be absorbed. So once we get to the brush border, you can see here a good visualization of the broken down fat droplets and moving it to the brush border here. So free fatty acids and the monoglycerides will move across this border via simple diffusion. across the apical enterocyte membrane right here. Um, so these move across via simple diffusion. You don't need an energy source to be able to do this. Cholesterol, however, moves across via specific energy-dependent membrane transporters. One example of this is NPCK1, and these are inhibited by enzymatic. I don't think I'm pronouncing that correct, but they're inhibited until the body is ready to absorb this cholesterol. Um, you see here the cholesterol and triglycerides and protein are all congregated in here. Um, the way that we get triglycerides, once the free fatty acids and monoglycerides move into the cell, they go to the smooth endoplasmic reticulum, and here is where they're recombined into their initial triglycerides. After that, the triglycerides are packaged with the cholesterol and the proteins, and these form large droplets of fat called chylomicrons. And you see right here a picture of the chylomicrons. You have phospholipid, the hydrophilic heads on the outside, the hydrophobic tails on the inside. You have proteins, cholesterol, and the fat droplets in the center. And you can see on the outside it kind of mimics the phospholipid membrane of most cells. You have the hydrophilic heads on the outside, and the phobic tails on the inside, and embedded in that is the proteins. So once these are ready to leave the cell and be absorbed into the body, chylomicrons are packaged by the Golgi apparatus and are leaving the cells in the secretory vesicles via exocytosis. So because chylomicrons are so large in size, they cannot cross this basement membrane into the blood. So you see here you have the chylomicrons packaged by the Golgi apparatus. They leave the cell. However, because of their size, they can't cross into the capillary. So instead, they enter into the lymphatic system through the lymph vessels of the villi called lacteals here. And through that, they will pass through the entire lymphatic system before they enter the venous blood, just before it enters the heart. So right into the vena cava is where it's dumped. Additionally, some shorter fatty acids are not assembled into chylomicrons, and they can just cross into the blood here through the basement membrane without needing to go through the lymphatic system first.